Thanks very much. Thanks, guys. Thanks for having me. So today I'll just run through a real basic overview of leukemia and then a couple of slides on what our lab do. But thanks for having me too. Really appreciate this opportunity. So just about me first. My mum's from Burma. My dad's from Poland. I was born in England. And as a result, you get me. So I grew up in Dio, so Diamond Creek. This is our landmark, which is a train station next to our historical McDonald's. So when I was at university, I was a welder in a factory in West Heidelberg and didn't really know what I was going to do. So I did a mathematics degree at Melbourne Uni with these people. And I didn't know what I was going to do. And then I met Ross Dickens, who's my current supervisor playing football at WEHI. I was cleaning dishes for a lady, Anne Verhagen. And next thing I knew, I was here reading the newsletter. So that's me. And now I'll go into actually some science. So what is leukemia? So leukemia itself was actually discovered as a disease and characterised in the 1800s by Dr. Peter Cullen in Scotland. And basically he described it as milky blood. So at that point, he didn't know why these patients were sick, but when their blood was taken, it was extremely white. And what you can see here is this is a blood sample from a healthy patient. They've spun it down using a centrifuge. You can see a lot of red blood cells, a little bit of white blood cells, and then this is serum where all your antibodies and everything will float around. But when you take blood from a leukemia patient and spin it down, you can see that the majority of blood in there is white blood cells, and these are not functional blood cells. So that's why leukemia patients present with things like they're anemic, so they don't make a lot of red blood cells, they can't respond to normal infections, and you'll see why in a minute. So until 1845, there was no real name for this disease until Rudolf Virchow coined the phrase leukemia, which basically means leukos is white, hema is blood. So that is why it's called leukemia. So when you actually take some blood of a healthy patient, spread it on a slide, and then look under a microscope, this is generally what you see. You see a bunch of little red blood cells here and a few mature myeloid cells. So these are things that respond to bacteria that you will encounter every day. But when you take blood from a leukemia patient, what you can see is a bunch of immature blood cells. These are the purple stained blood cells here, and they're really clotting up the blood, so you're not really producing functional immune cells anymore. So there's a bunch of different types of leukemia as well. Leukemia is not just one disease, it's a series of diseases. And the actual disease itself is derived from the bone marrow. You know, in a normal situation, Bandura. In a normal situation, a stem cell will produce all the red and white blood cells of your body in order for you to fight immune challenges and also to provide oxygen to all of your organs. So the stem cell can either produce what's known as myeloid stem cells or lymphoid stem cells. So these are sort of, eventually they'll become your red blood cells, platelets, which clot wounds, and myeloid cells. And for lymphoid cells, these are your more specific immune cells, things that can specifically target different pathogens, so bacteria, virus. And these all go through different stages. So they mature down these stages through stem cell state, through what's called a blast state. So this is a rapidly expanding point in development, and eventually they'll mature into the cells that circulate throughout your body. Now, the disease itself is really a disease of the more immature cell types, so your lymphoid blast cells, your myeloid blast cells. It can be either acute, so that is it's extremely immature and rapid in onset, or chronic. So people can live with a chronic leukemia for a long time and then eventually it goes bad. Once again, it can be either lymphoblastic, so down this line, or myeloid, so down this line. Now, there's four main types that are recognised. There's acute lymphoblastic leukemia, which is what I'll focus on today. It's fairly well treated, but you'll see there's still issues. There's a more common in adults is acute myeloid leukemia. So this is particularly poorly treated and something that's a major focus of a lot of groups around the world. And then you have the chronic diseases, which have been treated fairly well over time, and WEHI has a particularly strong tie to this chronic disease, chronic lymphocytic. So as I said, I'll focus on acute lymphoblastic leukemia today, which is a disease of these immature white blood cells, these lymphoid cells. So how does leukemia develop? And the way you can understand that is by trying to understand, well, what's the normal goal of B cell development? So a B cell is an immune cell that recognizes bacteria, viruses, etc., and produces antibodies that circulate around your body, allowing you to respond to secondary infections over time. So the goals are really to do this. It's to make B cells that can recognize foreign invaders, so bacteria, fungi, all these type of things, but also to make immune cells that won't actually attack your own tissue, because you don't want autoimmunity. You don't want things like MS, lupus, which is your body incorrectly killing itself. So how does the system do this? You have to make what's called a B cell receptor. Now what a B cell receptor is, it's a little protein that sits on the surface of the cell, and it actually scans the environment. So there's different proteins and antigens presented over time, and it will determine, is this a good one, is this a bad one? And the way you do that is through this B cell receptor. Now this receptor, like I said before, has to be to non-self, i.e. bacteria, etc., things that you're not normally interacting with. So from this blood stem cell, and I'm going to use horrible analogies throughout this, so I apologize in advance, but from the blood stem cell, like I showed before, you can make red blood cells, white blood cells. We'll focus on B cells here. So you have a progenitor stage here, which is the cells are getting more committed to becoming a B cell. And then you have the first audition of B cell development, which is this precursor stage. What that is that there's a massive burst in the amount of these precursor B cells, and that provides a whole bunch of cells which the body can then audition, if you will, to go to the next stage. So you need to have a bunch of diversity, then you pick out the ones that are best and they progress over time. So it's like Australian Idol. I'm not really up to date with the new singing ones. And to do this, you have to first ask the question, well, can you make this BCR, that protein that I explained before? Is it possible to do this? So you make a bunch of cells, and as I said, this little arrow means they're rapidly dividing, they're rapidly expanding and replicating their DNA, and this is prone to problems, as you'll see. Once this goes through, they have to say, well, are you actually able to make a functional BCR? So these are cells here that now they've progressed further along, their BCR is more mature, but now can it actually recognize bacteria and pathogens, or is it going to harm the cells itself, the body itself? And then over time, once this selection's happened, you have the winners, they circulate throughout your body, and they're the ones that are the best. So they're the ones that aren't going to kill you, but they're going to recognize foreign things. So this happens in the bone marrow, these stages, but this happens in the blood, the lymph nodes, and the spleen, which is why when you have mice that come down with leukemic sickness, you generally see things like splenomegaly, so enlarged spleen, enlarged lymph nodes, anemia. It's because this whole system is clogged up by bad guys. So leukemia itself, this is where the problem arises, when all the cells are dividing, and trying to make a lot of diversity to allow for that audition to go through. And what happens is you have to make a lot of DNA, it has to be replicated a lot, and the problem with that is that it's prone to genetic changes over time, because every time you make a copy of DNA, and also intrinsic in this process is you're chopping up bits of DNA to make that diversity, there's going to be problems with that over time, and some cells will have the wrong genes deleted just by chance. So this is what leukemia is. It's DNA changes that occur in this population that cause the uncontrolled growth of these immature cells. So these cells are not functional cells at all. They can't fight any infections. They're basically useless at this point. And what's happened is that you've had deletions of genes or parts of DNA that would normally control how well the cell can mature over time or stop it from expanding too much. So this is deemed the brakes of cell cycling. But also, as a result of chopping up bits of DNA, you may activate genes that promote the growth of cells. So you've basically put your foot down on the accelerator and taken off the brakes at this point, and you're getting too many of these cells. And there's too many duds, they won't stop dividing. And what that means is you get this excessive amount of immature white blood cells that spread around the body. They then clog up normal organ function, so in your spleen, in your lymph nodes. Eventually they may track to your central nervous system and just stop normal organ function. They also cause anemia because now the bone marrow is chock full of these, Therefore, you can no longer make red blood cells at a normal rate, so the patient won't be able to make these cells that transport oxygen around the body. So if I'm not explaining this correctly too, just let me know and stop me as I go. So who's affected by this disease? Acute lymphoblastic leukemia, so ALL. Really, it's children that are the main victims of this disease. It's the most common cancer in kids, and it's also the leading cause of cancer-related death in children. But as you can see, there are some adult patients, and this is from an American population, but you can see there are some adult patients that do get this disease, and they're particularly important, as we said. So how is this disease treated? Well, classically, the disease has been treated with a series of approaches, including steroids, which immature white blood cells are particularly sensitive to steroids. They undergo cell death or apoptosis when you treat with steroids. There's something called genotoxics, such as this drug vincristine, and that just kills rapidly dividing cells. So when you see patients who have no hair, have skin and gut problems, it's because the drugs aren't specific to cancer cells, they're specific to cells that just divide a lot, which is also cancer cells. But it's also hair progenitors, it's also gut lining. These are all cells that rapidly divide. And then if these don't go so well, and classically they've done pretty well over time, you go for a bone marrow transplant. That's really risky. Hopefully you have a sibling that is matched to you or a donor that is matched, but that's just a way to kickstart the blood system. The problem with all of these, and because they're so non-specific, there's a lot of side effects that happen. So patients have problems with bone development, they can develop diabetes, behavioural issues, because this is a steroid, so you're throwing out of whack the normal hormonal balance. There's gut toxicity, like I said, you're killing the gut lining. Immunosuppression, so even though you're killing cancer cells, doesn't necessarily mean you're making cells that can respond to bacteria yet, so these patients are immunosuppressed, and they don't make platelets, so they have clotting issues. So really, the name of the game now is to make more targeted drugs that are only available at the moment. There are certain drugs that do treat certain patients, but we need far more drugs to specifically kill the tumour cells. So not all this collateral damage anymore. We want drugs that will actually target the tumour cells themselves. Historically, though, there's been different responses to the drugs that I presented. So for children, for example, in the early 1960s, if you had acute lymphoblastic leukemia, your chance of survival was extremely low. However, nowadays, they're looking at 9 out of 10 patients will respond really well to the drug. So it's really good for children, these drugs. They respond really well. This is a child here who's holding up a poster saying that even though he gets chemo in the afternoon, he can still play sport. So kids respond quite well to this disease with 80 to 90% survival. But there's still that 10% that we don't know what to do with. So it's really 1 in 10 is still not a great statistic over time. If there's 1 in 10 patients that aren't going to get through, it has to be improved. But more harrowing is perhaps the fate of young adults. So these are American patients here in a particularly high-risk subset. So you see that children aren't as good as I said before, but these are a group of patients that don't respond at all to drugs really. And you can see young adults are particularly poorly treated, and collectively they respond with around 30 to 40% survival rate. So that means that there are now 6 out of 10 patients that won't respond at all. So we're trying to understand these real key questions here. Firstly, why do these patients relapse? So the 1 out of 10 in the kids and the 6 or 7 out of 10 in adults. Why do they relapse? Why are adults so poor at responding to chemotherapy at the moment? And then also, what are the genetic changes that cause a normal white blood cell, so a normal progenitor cell, to then be transformed and become a tumour? And then how can we attack these mutated cells, and particularly those that can't be killed by chemotherapy at the moment? So it's all about enhancing specificity of treatment at the moment. So to go into the last little bit of this talk, we just have to understand the normal DNA to protein. So you can consider DNA itself the control center. So this has the imprint to do all of the functions in a cell. However, within a given cell, there are different messages that are passed on to the cell to produce the product and then the function. So imagine it's like a cookbook. You're not going to cook every recipe in the book. Every cell uses a different group of recipes to produce the function. So therefore, this is a really critical dogma. If we can understand what happens in cancer to this process, then we can potentially understand what we can do to treat these cells. So the way you do this is you want to look at the genetic makeup of ALL patients here. First thing you can do, and this isn't very clear, but we have a group of patients here and then a group of people that don't have leukemia. And basically we compare the DNA from the patients to the healthy people and say, well, what are the genes that are either mutated or deleted in leukemic patients versus healthy individuals? So what is giving these people a predisposition to developing leukemia? That's one way to do it. Another way is to take a patient themselves, take tissue at diagnosis. So when they come down with leukemia, you take diagnosis tissue and you compare the DNA of the diagnosis tissue to that of normal tissue, and you say, well, what's the changes in the white blood cell that make it different from every other cell in the body that's healthy? And then finally, what you can do is you can look at the relapse. So these are the patients that haven't responded. You can take tissue at the diagnosis stage, look at the DNA here and compare it to the DNA that's occurring at the relapse stage. So you take the cells at relapse and just compare relapse to diagnosis. So this is what, over time, and this is many studies have done this, and not our lab in particular, but we actually became really interested in this question of a gene called Icarus because it fits this criteria. So there are mutations in the gene Icarus that result in it no longer having function, so it's deleted, that are enriched in leukemia patients compared to healthy people. So it suggests there that Icarus deletion is a predisposition to developing a leukemia. Another thing they noticed was that Icarus is actually deleted in the leukemia cells, but not healthy tissue of these patients. Then finally, if you compare the population of cells at diagnosis to those that grow out at relapse, actually you're more likely to have an enrichment for deletions in Icarus at relapse of disease than diagnosis, suggesting that cells that have a deletion in Icarus are particularly resistant to chemotherapy. And this is shown here. So what I'm showing here on this y-axis is the incidence of relapse, so how many patients actually did not respond to chemotherapy. For these patients, they have normal Icarus function, so they respond fairly well to chemotherapy in this group. However, when Icarus is deleted, your chance of relapse is extremely high. So what it suggests is that with an Icarus deletion, there are currently no chemotherapeutics that are able to treat these particular patients. So that's why we became quite interested in this. And to do this and to study it, Ross, my boss Ross, went to Cold Spring Harbor in New York and basically learnt a really cool technique at the time, which was known as RNA interference. So if we go back to DNA to RNA to protein, as the name suggests, RNA interference, what you're doing is if you can get rid of the RNA here, so the message, you'll no longer produce the function and you can disable the function of your gene of interest. So for us, it's Icarus. We know that patients that have a deletion in Icarus have no longer any functional Icarus protein. So if we knock down the expression of Icarus here, so knock down the target gene expression, we get knockdown of the protein Icarus expression. And this entire process was found by Andy Fire and Craig Mallow in 2006. They won the Nobel Prize for this. So the name of the game for our lab is really to knock down genes of interest in white blood cells to mimic what happens in a human. In a human, you have a deletion of Icarus, then in a mouse, we're able to knock down our gene of interest. But one of the best things about using this system is it's completely reversible. So even though you knock this down, that isn't forever. You can actually switch the gene back on over time, and that's why we get Ketty to put mice on docs or this and that. Docs is actually the trigger to switch the gene on and off in our system. So what I've done throughout my PhD and honours was to generate mice where we could knock down the expression of Icarus in white blood cells of a mouse. And then we actually cross these mice with mice that are prone to developing a leukemia to see whether or not if you lose Icarus plus have a genetic predisposition to leukemia, you see any interaction between these. So what you can see, and this animation has not worked in any way, basically, this hasn't worked at all, but what you see is with Bcell alone mice, they develop a leukemia around 100 days post-birth. However, if you knock down Icarus in combination with that, they develop a leukemia at around 50 days post-birth, suggesting that Icarus is actually disabling the tumour suppressive function of a cell, i.e. it promotes the onset of leukemia. So Icarus loss plus Bcell is bad news for a mouse. So what we were then able to do is to take these cells and transplant them into recipient mice and utilize that switching on and off that I discussed earlier. So you take the leukemia and put them into rag mice. The reason we do this is rag mice don't have any mature white blood cells to actually knock back this transplant. And then you can either leave them untreated, which means that Icarus is off, as it was when it went into the mouse, or when the disease comes up, we treat with dogs and we switch Icarus back on. So this is what happens. You transplant the cells, Icarus is off in these cells, the mice succumb to the disease after transplantation at around 30 days. However, if you switch Icarus back on in the tumour cells, what we find is that these mice actually have a significantly increased survival, and for a couple of mice here, they've been completely cleared of the disease, so they're still healthy and on the shelf today, suggesting that these cells depend on Icarus loss for their survival, because when we switch it back on, they don't like that very much at all. Now, this is what it looks like in the blood of a mouse. So what I'm showing here is FACS plots, which is exciting to me. So when you transplant these into mice, this is at the initiation of sickness. So what we look for here is they might be scruffy, have splenic enlargement, all these type of phenotypes. If we don't treat these mice and we keep Icarus off, you can see here, this little red box is the tumour cells circulating in the blood. Over time, these cells grow out and eventually the mice will succumb to the disease. However, if we take a mouse here and we switch Icarus back on, you can see that the tumour cells are no longer circulating in these mice. So the mice are completely healthy and they were dependent on the loss of Icarus for their survival. So what we've identified now is a gene that's critical for the survival of tumour cells, and we want to look at why that is over time. So that's what I'm doing now. We're looking for genes, and this is the last slide actually, I've run out of time here, but we're looking at genes that when you lose Icarus, what genes in the network of genes that are expressed in the cell are dysfunctionally expressed in these tumour cells compared to patients that don't have an Icarus mutation. So what we do is we use the mouse and we use human data and we combine the two. We're trying to look at conserved pathways between human and mouse that might give us some clues into new ways to treat patients that have an Icarus mutation. And genes that are really highly expressed in Icarus deleted patients are of interest because the tumour cells may depend on them being extremely highly expressed for their survival, and they're potentially druggable. So really the overall aim of my work and the work of many groups around the world is to improve the health of these Icarus mutated patients, because at the moment they have very few treatment options. So that's where I'll leave it today. If there's any questions, I just want to thank a few people. So I want to thank Kenny. I've known everyone in the mouse rooms for a long time. but I really appreciate. All right, calm down. So I want to thank Katie and Carolina, who are in the room at the moment. They take care of all my mice. I want to thank Emma and Mel, who are former managers of our lab. All the people mentioned here and everyone else that helps out with our mice, particularly people on the weekend that come in and check our mice and let me know. We really appreciate it. But yeah, thanks to everyone for all your help with all of my work and making this a really fun place to work. I'm leaving soon, so I really appreciate it. Cheers, guys. Thanks. Questions? Like that, bro?
Do you know why it's higher in children?
Yeah, so there's a few ideas out there. So firstly, the immune system itself is really kicking off in kids, so that's where all that progenitors are all growing up and being selected. But over time, that whole system starts to settle down when you produce a repertoire of cells in your body that circulate. So really, it's a volatile time in development of any human because you're starting to boost all the white blood cells you're making to set a human up for the rest of their life. There's a bunch of ideas out there like radiation. So really the biggest human experiment ever conducted was actually atom bombs in Japan. So what that did was it presented a population that were subject to radiation over time. What we're seeing now is a bunch of patients that are coming down with leukemia or lymphoma from Japan. And what that suggests is that we can look at the genes that are deleted in these patients and say, well, these are the ones that are subject to an environmental insult. So these are the ones that cause cancer. But yeah, in kids, it's just because you're starting to kick off. Hey.
You said this is mainly in the ALL leukemia, but is this gene of a big interest in the other types of leukemia as well?
Yeah, so in AML, it's also of interest a little bit. So in AML, if I go back to one slide, it's a really good question. So there's a few diseases out there that Icarus is important to. So here, there are a few diseases called multiple myeloma, which is actually a disease of mature blood cells. Now, Icarus itself is normal in these cells, but if you target it with a drug, just Icarus protein, you can actually kill multiple myeloma, where here, if you target it, and by knocking down Icarus, it causes cancer. So it's a bit of a double-edged sword there. However, in AML, which can develop here, so AML is a disease of myeloid cells that are immature. What happens in cases of chronic myeloid leukemia, so these are patients where they can sit there, they can have leukemia for quite a long time, but eventually they just explode with an acute disease. And what they found is that acute disease, so the really rapid, the one that causes sickness and for a patient to go into a clinic, the reason why these patients seem to come down with it is because the cells acquire a deletion in Icarus, they go on to develop a really bad disease. But before that, Icarus was normal. So it has some kind of function in myeloid disease as well. Great. Thanks very much, guys. Cheers. Thanks so much.